In the name of Allah, the Beneficent, the Merciful. PCR applications include mutational analysis, paternity testing, RFLP analysis, confirmation of diagnosis for certain infections, forensic analysis, and detection of subclinical infections. PCR is a technique in which selected region of chromosome can be amplified more than 1 million-fold within a few hours. It allows extremely small samples of DNA to be used for further testing. The region of a chromosome to be amplified by PCR is referred to as the target sequence and may be an area containing a suspected mutation, a short tandem repeat, a microsatellite sequence, or any area of interest. PCR can amplify even if the target sequence makes up less than 1 part in 1 million of the total sample. To perform PCR, it is necessary to know nucleotide sequence of target DNA or the nucleotide sequence of short segments on each side of the target DNA. These stretches are called the flanking sequences, which bracket the DNA sequence of interest. Along with that, 2 oligonucleotide primers are required, as well as sufficient quantities of deoxyribonucleotides are needed, along with heat-stable DNA polymerases. The primers are constructed as 2 single-stranded oligonucleotides, 20 to 35 nucleotides long, which are complementary to respective flanking sequences. The 3' OH end of each oligonucleotide points towards the target sequence. The target DNA is heated to 95 degrees centigrade for strand separation. The separated strands are cooled to 50 degrees centigrade and primers are allowed to anneal to the complementary sequences on the separated strands. And DNA polymerases are allowed to continue and elongate the chains. So here you can see chain elongation by DNA polymerases. DNA polymerases and deoxynucleotides are added to mixture at 72 degrees centigrade for strand elongation. So the green colored are the primers and the red colored are the deoxyribonucleotides, which are being added to the primers for chain elongation. So that is how the DNA strands are being duplicated. At the completion of one cycle of replication, the reaction mixture is heated again to separate the strands. Each strand binds a complementary primer. Step of primer extension is repeated by using heat-stable DNA polymerase, for example, TAC from Bacterium thermus aquaticus that normally lives at high temperatures. TAC lacks proofreading activity. 20 to 30 cycles are run and DNA is amplified millionfold to a billionfold. Each extension product includes a sequence at its 5' end that is complementary to the primer. Thus, each newly synthesized strand can act as a template for successive cycles. This leads to an exponential increase in amount of target DNA. Cyclic heating and cooling carried out by computer-controlled thermostat. After only the third cycle, double strands start to form with a length equal to the distance between the primers. The proportion of these doubles during each cycle until all strands have correct length. Probes can be constructed during PCR by adding labeled nucleotides to last few cycles. Now coming to the applications of PCR. The Huntington disease is caused by a trinucleotide repeat expansion in the coding region of the gene, which means many extra nucleotides are present in multiples of 3 or longer. DNA fragments may be expected in case of this mutation. This can be confirmed with the help of PCR because mutation changes the length of the gene, for example, microsatellite repeats, and this difference can be detected in the PCR amplified DNA by electrophoresis on agarose gel. PCR amplification is carried out on regions suspected to contain the mutation. In Huntington's disease, a trinucleotide repeat expansion of CAG codon in exon 1 of the gene occurs, which is for the Huntington's protein. It leads to insertion of many extra glutamate residues in the protein, causing neurodegenerative disorder. The additional glutamate residues result in abnormally long protein that is cleaved, producing toxic fragments that aggregate in the neurons. So here you can see the green colored extra glutamate residues. Coming back to the question, the results are consistent with Huntington disease and in comparison with the normal PCR of the control. The 170 base pair long fragment of patient's DNA is well out of normal range. Not only that, it is also consistent with the triplet repeat expansion in that allele of Huntington gene. The two bands from the control are 95 and 101 base pairs. 101 minus 95 is 6, which equals to 2 trinucleotide repeats. The patient's sample shows a 104 base pair band, a difference of 3 base pairs or 1 nucleotide repeat from the 101 base pair band of control. 170 minus 104 equals 66 plus 1 nucleotide repeat, which has caused the longer fragment to be 104 base pair instead of 101. So it makes a total of 69 base pairs, or 69 divided by 3 makes 23 nucleotide repeats, which clearly reflects trinucleotide repeat expansion. So here you can see that blood donors are screened for hepatitis C virus by anti-HCV antibody test through chemiluminescence, ELISA or EIA. In case of positive result on ELISA, the HCV RNA genome testing with the help of PCR-based technology is a confirmatory test for HCV infection. So now this is a case of trinucleotide repeat expansion in non-coding region. In this case, trinucleotide repeat expansion occurs in an untranslated region of a gene that the result can be decrease in amount of protein produced. It is the most common cause of intellectual disability in males. The expansion results in gene silencing through DNA hypermethylation. Hypermethylation leads to gene silencing because methyl groups serve to recruit histone proteins. This leads to tight packaging of DNA into heterochromatin, leading to failure of gene transcription through epigenetics. Fragile X mental retardation 1 or FMR1 trinucleotide repeat expansion on 5' site in untranslated region of the FMR1 gene provides instructions for making the FMRP, which plays role in development of synapses in brain. Patients with Fragile X syndrome do not make FMR1 protein of gene because of gene silencing through hypermethylation. This trinucleotide repeat expansion will alter the length of the restriction fragments. Since this man is suspected to have Fragile X syndrome, for confirmation of diagnosis, the loci likely to be involved are amplified by PCR and further analyzed for mutations. Since mutation for Fragile X syndrome may cause a length difference in the PCR product, the length differences can be detected by gel electrophoresis of the PCR product. Previously, the DNA at crime scene was used to be analyzed for DNA fingerprinting by RFLP analysis. The purified DNA was cut at specific points with help of restriction enzymes. The fragments of different lengths which were obtained were placed on a gel, and the gel was subjected to an electric current for electrophoresis. The shorter the fragment, the more quick used to be its movement towards the anode. The sorted double-stranded DNA fragments were then subjected to a blotting technique in which they were split into single strands and transferred to a nylon sheet. These fragments were subjected to autoradiography in which they were exposed to DNA probes, which are pieces of DNA complementary to mini satellites DNA and that were made radioactive. A piece of X-ray film was exposed to the fragments. A dark band used to be produced on the X-ray film where radioactive probes were attached. The resultant pattern of marks was analyzed, but that was a cumbersome procedure. This assay has been supplanted by approaches based on PCR and microsatellites or short tandem repeats, which are 2 to 4 base pairs in length compared to mini satellites that used to be 10 to 100 base pairs in length. PCR amplifies the desired fragment with a specific STR many times over, creating thousands of copies of fragment. An exact sequence of nucleotides can be determined by DNA sequencing. To detect Borrelia burgdorferi DNA in a patient's blood, a probe complementary to bacterial DNA is hybridized with target DNA and detected by Southern blot technique. These fragments are subjected to autoradiography in which they are exposed to DNA probes, which are the pieces of DNA complementary to bacterial DNA and that were made radioactive. A piece of X-ray film was exposed to the fragments. A dark mode produced on X-ray film would, with radioactive probes attached, would show the presence of the bacterial DNA, and the resultant pattern was analyzed. This assay has been supplanted by approaches based on PCR. PCR amplifies the desired segment with specific bacterial DNA many times over, creating thousands of copies of the fragment, and exact sequence of nucleotides can be determined by DNA sequencing. PCR offers a rapid and sensitive method for detecting viral DNA sequences even when only a small portion of cells harbor the virus. Quantitative PCR or qPCR, also known as real-time PCR, allows quantification of the amount of DNA with each cycle of amplification, and that is in real time. It is useful in determining the viral load. For screening, anti-HIV1 and 2 antibodies are tested through enzyme immunoassays or chemiluminescent immunoassays or ELISA. AIDS diagnosis can be confirmed by PCR. Severity of AIDS can be checked by CD4 count. The enzyme-linked immunosorbent assay, ELISA, is used to screen individuals for antibodies to the HIV virus. This test has high sensitivity but somewhat lower specificity. A positive result in ELISA must be confirmed by a Western blot for antibodies that are reactive with specific HIV protein antigens. For PCR, primers specific for HIV provirus are used. If person is infected, the proviral genome will be amplified and detected. If person is not infected, there will be no PCR product detected. It has two advantages. It is much earlier after infection and does not rely on an antibody response by the individual. Reverse transcriptase PCR or RT-PCR detects and quantifies specific RNA. It is useful in detecting RNA viruses. A blood sample from HIV-infected individual is obtained and after appropriate preparation is treated with reverse transcriptase to produce complementary DNA from any RNA in the sample. The complementary DNA is subsequently PCR amplified using primers specific for end sequences of HIV complementary DNA. The amplified product is quantitated and with use of standard curve, it can be related to the original amount of HIV RNA present. To detect HIV infection in a baby born to an HIV positive mother, PCR is performed. Primers specific for HIV provirus are used. If newborn is infected, the proviral genome will be amplified and detected. If newborn is not infected, there will be no PCR product detected. It has two advantages. It is much earlier after infection and it does not rely on an antibody response by the newborn. The sample for suspected COVID-19 infection is obtained and after appropriate preparation, it is treated with reverse transcriptase to produce complementary DNA from any RNA in the sample. Complementary DNA is subsequently PCR amplified using primers specific for end sequences of the COVID-19 complementary DNA. The amplified product is quantitated and with use of standard curve, it can be related to the original amount of COVID-19 RNA present. For a couple heterozygous for beta thalassemia mutation, with a son who is homozygous for the normal allele, one can conclude that two restriction fragments shown in this pattern are derived from chromosomes without mutation. It is also clear that the upper fragment came from the mother and the lower fragment came from the father's chromosome. The fetus has fragment from mother's normal chromosome. The other fragment, which is the top one, top one on the blot, must have come from the father's chromosome with mutation. The fetus is therefore heterozygous for the mutation. A is the son of parents 1 because PCR product on blot match M1 and F1. B is the son of parents 2 because PCR products on blot match M2 and F2. PCR can amplify the sequence even when the targeted sequence makes up less than 1 part in a million of the total initial sample. DNA samples from victims are taken from tendon, arm muscle tissue, bone parts, or parts of the molar tooth. The DNA sample from plane crash victim is compared with DNA of family or DNA obtained from unwashed clothes at home, unwashed collars, or toothbrushes, etc. The process of creating a DNA fingerprint is first DNA is extracted and purified from sample, which may be muscle tissue, bone tissue, or dental tissue. In this case, for RFLP analysis, the purified DNA is cut at specific points with help of restriction enzymes. The fragments of different lengths which are obtained are placed on a gel and gel is subjected to electrophoresis. The shorter fragment, the more quick is the movement towards the anode. The sorted double-stranded DNA fragments are then subjected to a blotting technique in which they are split into single strands and transferred to a nylon sheet. These fragments are subjected to autoradiography in which they are exposed to DNA probes and these probes are radioactive. A piece of X-ray film is exposed to the fragments. A dark mark is produced on X-ray where radioactive probes are attached. The resultant pattern of marks is analyzed, but this is a cumbersome procedure, and this assay has been replaced by approaches based on PCR and microsatellites, which are short tandem repeats, 2 to 4 base pairs in length, compared to mini satellites that are 10 to 100 base pairs. PCR amplifies the desired fragment with a specific STR many times over, creating thousands of copies of fragment, and exact sequence of the nucleotides can be determined by DNA sequencing or NAT, which is nucleic acid testing. Here the answer is C. Here again the answer is C, which is microsatellite tandem repeats.